So I just want to make two observations. We're going to go from two incredibly dedicated and talented researchers talking about scales to two other incredibly dedicated and talented researchers talking about EEGs. And you're like, Mike, what happened? You just threw it all this was like your leftover category? And the answer is no. I've been doing this for 5 years. And for 5 years, every time I talk to industry, they look at me at the end of our conversation and say, what are we going to measure in a clinical trial? And it was the bang your head against the wall question. And this session is the answer. We have Tom Frazier developing something that's already got 2 publications behind it. We have Christy being funded by the FDA to do a scale that we can use with the FDA. And now we're going to hear from 2 clinicians who will hopefully help us find some signal in those squiggly lines that might also be a signal. So this is the holy grail, guys. We know we have scientists, we know we have good work happening, but we have to get these therapies through trials, and we get through trials by having something we can measure that changes. So this is incredibly exciting, and I'm thrilled to be introducing our next 2 speakers. Just in the interest of time, I'll let you talk, and then I'll let you pass it to the next one. And I'll ask them to introduce themselves, because I'm obviously terrible at introducing people. But stay with us. I've been waiting 5 years to have a talk about this. So with no further ado, all the way from Edinburgh.
Wow. Thank you, Mike. Yeah, that's the best introduction I've ever had by miles. Thank you so much. It's really an honor to be here. I'm from Edinburgh. So nice to be in Florida, in the sun. I've learned so much today. The science has been amazing, but also really from the families. Just these interactions are really important. I'm actually not a clinician. I'm a preclinical scientist. I mainly work on rodent models. And yeah, seeing what the truth is with people is really, really critical. So I'm going to tell you about our data. We work with rats in Edinburgh. The idea behind rats in this research is that they may have more complex behaviors than other rodents, and so we can study these. And the rats that I'm going to tell you about were developed by my collaborators, Peter Kind and Sally Till. Sally was here last year telling you about the behavioral phenotypes that they see in these rats, which are issues in learning, and also in the ways that they socialize. Quick step back, these rats have their gap domain and C2 domains knocked out. There's another version of this model where the entire gene is knocked out, and the phenotypes that I'm going to tell you about in terms of seizures and sleep are similar in both of these. So my lab is really specialized in vivo EEG recordings in rodent models of epilepsy and neurodevelopmental disorders. And I was quite envious of the talks earlier with frogs and flies. So these are hard animals to deal with. These are basically one-man brain surgery experiments that are very, very long to do, these surgeries, 4 hours usually. And then we do long recordings, often over weeks, to get these data and to get these replicates. And so for these particular data, we're using these grids that sit on the surface of the skull of the animals and are permanently implanted to mimic human EEG and to look for biomarkers, which was what Mike alluded to. So very talented postdoc in the lab, Ingrid Buehler-Peralta, did these recordings. And you can see in that recording that kind of like 2/3 into the data from left to right, these kind of spikes and waves. And earlier we saw data from the SLC 6A talk that there were some absence seizures. And so these are absence seizures in rats. So one parameter that's really important in EEG data is these kind of waves that you see over time, and how fast do they occur, and how broad they are is important in finding seizures, but also just kind of normal activity. And so these are spectral analyses that we're doing. And so you can see the seizure. It's called the EEG like that is called a spike and wave discharge. So there's these big downward peaks. and then kind of a slower wave, and it's very repetitive over time. And when this is happening, the rat freezes, and it has this very slight head bobbing, which in this data, you can see that at the bottom, there's an accelerometer channel. So this is basically the same kind of device that's in your cell phone that measures where the cell phone is in space. And when we see the peaks of the seizures, we see this kind of tiny head bobbing. So I think it's a kind of like an eyelid myoclonia, which is one of the common seizures. I think that they also have reflex seizures. So often they'll be chewing, and you'll see the kind of chewing artifacts on the EEG, and then they'll go into a seizure, and sometimes they come out of the seizure, and you see the artifacts again. But it's quite a hard experiment to prove for sure that these chewings are causing the seizures. So we see them in our rats, and so one of the missions of this talk was to try and convince you that this is a good model, and all these therapies that we've seen today, potentially we should test, or at least some of them, test in this model. So compared to the wild-type siblings, are heterozygous knockouts, so to mimic the haploinsufficiency. So in this data set, 9 out of 12 of those rats displayed these spike and wave discharges, and only 2 of the wild types displayed them. But when the wild types displayed them, the numbers were much smaller, which you can see in that kind of middle quantification. And then on the right, you have this cumulative probability distribution, where you have on the horizontal axis the duration of the spike and wave discharges. And you can see in blue, so in the mutants, that's shifted to the right, meaning that they're longer. So these spike and wave discharges are longer in these rats, which, so when you have atypical absence seizures in patients, these are the ones that are harder to treat with drugs, and they are longer. So we think that they're a good model for the seizures. So rats are not tiny humans. They have many differences in terms of their behavior and their physiology, and one of the major differences is their sleep. So for all of you that have cats, they're kind of like cats. They take naps all the time. And they're also nocturnal. So they're more awake at night. And so you can see that in these plots. So each one of them is a different brain state that we quantify by the EEG. And on the horizontal axes, you can see the time of the day. And there's a white bar and a black bar in these data, and that's when the lights are on in the animal facility in the white, or when they're off in the black. And so binned over the 24 hours is the minutes that they're spending in each state. And so on the top one, you can see how much they're awake, and so they're normal in that sense, these animals. They seem to be more awake when the lights are off in the animal facility. Non-REM, which is often called slow wave sleep, and it's in humans, this is split up into various non-REM substates. We just kind of use one big quantification of that, and it's the majority of time that they're asleep. We see that more during when the lights are on in the animal facility. And then at the very bottom, you have REM sleep, which is often associated with dreams. So if people are woken up during REM, they report that they're dreaming. And you can see here that that's the big phenotype that we found in these animals, that their minutes that they're spending in this dream REM sleep is lower. And so this is where it got really encouraging for me. This is hot off the press data from my collaborator, Lindsay Mizzen, who's a psychiatrist in Edinburgh, and performed these very valiant EEG recordings going to patients' homes in the UK. And I thought that it was very clever what she did, which is to get control data from siblings of the Syngap kids in the same homes, in the same nights that they were performing the EEG. And so there's a lot of data associated with this. None of it is mine, and she gave me permission to share it with you. She did it with her colleague Stevie Williams, and basically similar finding as in terms of the REM. So they have decreased minutes spent in REM. Okay, so now to the title of the talk. And this is really all the work of my PhD student, Melissa Fassel, who, despite it being about 10 o'clock at night in Edinburgh, is watching on Zoom. And she's in the midst of writing up her PhD thesis, which is a kind of stressful time in life. And so she's very active on Twitter. So if I could get some people to follow her and encourage her, that would be great. So that's her handle on the bottom. And so the questions that she wanted to ask in this final stage of her PhD is, well, can machine learning identify SYNGAP1 individuals? And this is both actually in humans and in rats from their EEG. And what are the most important features to try and figure that out? So this is maybe not so important for clinical development of therapies or for biomarker, but for me that I'm a basic scientist and want to try and figure out why this is happening to the brain, then we can develop questions based on that. And are these same features really, do they extend across the two species? So there was the very nice talk of Gemma earlier where she was using machine learning, and we use quite a similar method. This is a tree-based approach, so basically ensemble learning, meaning that there's many variables, and on its own, one of the variables may not be as indicative as when they are grouped. And so essentially the machine learns, so it's supervised learning, so basically we'll feed it data where we've told it this is EEG data from a wild-type individual, a normal individual, or a SYNGAP individual. And then it'll kind of try and pick out what's going on. So this is the pipeline that, in terms of the analysis, it includes the data collection on the right. So EEG data is often very dirty. There's lots of artifacts, movement artifacts, electrical artifacts, so there's a lot of pre-processing that goes on. And what we do is that actually, well, I'll get to that in a second. So we break down all our recordings into small epochs. So this is not an aggregate of the entire 24-hour recordings, but actually in the rats, it's very small 5-second epochs of data. And in the human data, it's 30-second epochs. And so for each of those epochs, we perform lots of analyses to get numbers about that data. And these analyses are features from the data, and they look at different aspects of the data. So how complex is this signal? So there's analyses that figure out how repetitive is this voltage trace over time. Another set of features are these kind of spectral features. So meaning at what rate are the peaks and troughs of the data kind of coming in, and how big are their amplitude. And then another set of features at the end are functional cognitivity. So we have lots of channels, and so how correlated are they, are the signals? So how much do they interact with each other? So these are the different kinds of metrics that we're getting from the data, and then we feed them into the model. There's training, validation, so that'll be one set of data, and then we'll test on another. There's other aspects that maybe aren't that important for now, but there's something called hyperparameter tuning. So for the machine learning, you can tell the machine how many times do you go through the data. If you're building these trees, how many branches will they have? How deep, essentially, do you go into the data? And then we test. So this is the splits of the data, meaning for our rodents. So this is so we use 10 of our animals from the controls and the mutants to train the data, and then we test on 5 at the end. So the machine, for reasons that I don't really fully understand yet, will actually kind of pick up signatures from the data and will learn it in such a way that if you train on the same individual that you test, it'll know that, and it's kind of cheating. So what we do is to only test and train on separate groups of individuals. But as Gemma actually stated during her talk, this kind of modeling really needs loads of data. And so we're breaking down all of our recordings into these individual epochs, and those are the numbers that you see. And we'll test our accuracy, there we go, on these epochs. And so there's an aptly named confusion matrix on the right, which gives you how accurate the data is. So when you have like, when is it true that something is not a SYNGAP animal, and when is it positive? So this is where we evaluate. And overall, in our rodent data, the numbers are pretty good. Those numbers are decent for this kind of machine learning approach. So our accuracy and precision, they're at about 3/4 of the data. And remember that this is at individual epochs of time. So then what we can do is to do something called majority voting, which means that for our individual, we can see how frequently was it called a SYNGAP individual or a control, and take the majority, and that allows us to identify them as being a mutant or not. For the human data, we have similar types of splits and tests, and it seems to be even better. So the accuracy is more hovering towards the 80% range in terms of the human data. So it's really quite encouraging. And in terms of feature importance, so the type of machine learning that we're doing, which is something called XGBoost, there's analyses that let you go back into the machine learning and figure and see kind of readouts of what the main metrics were that or features were that it allowed it to identify. And the ones that seem to be the best or kind of are always highly ranked are the ones that involve connectivity. So CC on the left there is cross-correlation between channels. We also have some of the other features in terms of kind of gamma, which is a frequency band, so this is kind of a spectral analysis. But it seems that connectivity overall is really critical for the machine to be able to identify. And in the human data, it was the same. So there we have the cross-correlation again on top, as well as phase locking, which is another measure of how synchronized the different EEG channels are. So conclusions for the talk are, I think that our animal model is really good. So it displays these absence seizures that are very prevalent in SYNGAP patients. There's also REM sleep deficit, which really, I'm still kind of so happy every time that I see this data that we also see it in the humans. And this is a relatively new technique that Melissa has been applying, and already she's gotten it to be at a quite high level of accuracy. So that's really nice. And connectivity is really the critical kind of feature to let us identify. So future work. So again, as a preclinical scientist, I really want to look at the underlying mechanisms. So SYNGAP is involved with the synapse, but as we've learned today, there's all these other possible functions, such as kind of structural properties where it's involved. So I think that it makes sense that there's issues in how the different regions of the brain might be communicating with each other. And then, of course, as Mike alluded, it's this idea of biomarker development. The data sets were, so there was one question from the previous talk about having different formats of data. I mean, they were very different, the types of recordings that were done, and it seems to have worked. So kind of what we're going to do now is to dig more into it. Can we mix up the data in different ways and get similar results? Could we, Peter actually suggested this, which is can we train the data on animal data and then test it on human data and get it to be as accurate? And so these are the kind of things that we're going to test now. And that was kind of my second mission of coming here as well, was to try and potentially get some EEG data that you guys might have, and that might be possible, it seems, from talking to different people. So yeah, so just to thank the team, Ingrid that did the recordings, Melissa that has done this fabulous analysis, my collaborators Peter, Javier Escudero. So I'm a simple biologist, and I've gotten a lot of guidance from my collaborator, who's the second supervisor of Melissa. He's a signal processing expert at the School of Engineering in Edinburgh, and Lindsay and Stevie, who collected the human data and then did the counts of the sleep. Okay, so I think probably not enough time for questions. So.